السلام عليكم ورحمة الله وبركاته طلابنا الأعزاء، أسأل الله لكم تعالى الصحة والسلامة والأمان في ظل هذه الظروف اللي يمر بها بلدنا العزيز. محاضرتنا اليوم طلاب راح تكون عن البودي فلويدز. Body fluids. Body composition. In the average young adult male, 18% of the body weight is protein and related substances, 7% is mineral, and 15% is fat. The remaining 60% is water. That means 2/3 of the body weight is water, which is classified into intracellular fluids and extracellular fluid. The distribution of this water as the intracellular component of the body water accounts for about 40% of the body weight, and the extracellular component for about 20%. Approximately 25% of the extracellular component is in the vascular system, which means in the plasma. That's equal to 5% of the body weight, and 75% outside the blood vessel interstitial fluid, which is equal to 15% of the body weight. The total blood volume is about 8% of the body weight. As we see in this figure or the diagram, the components of the body and how the fluids is distributed between these compartments. Also, this figure showing the equilibrium of fluids through intake of water and the output of it through the lungs, kidneys, feces, sweat, and skin. Forces producing movement of substances between compartments. Number 1. Simple diffusion, in which the molecules or ions tend to separate from region to another region through the membrane lipid bilayer for lipid-soluble substances such as oxygen, nitrogen, carbon dioxide, and alcohol, or through the membrane pore, which is nonspecific holes in the lipid bilayer and include movement through the hydrophilic centers of protein complexes, or protein channels for water and water-soluble substances such as ions and glucose, until the concentration is uniform across the membrane. It does not require metabolic energy and therefore it is passive. Diffusion from one region to another is affected by some factors. Before that, dear pupil, in this slide we mean the diffusion of substances according to the chemical or electrical gradients. That means the transport of substances occurs from the region of high concentration to the region of low concentration. That affecting the simple diffusion are A. The concentration gradient, B. The electrical gradient. Whenever there is a difference potential between two regions, positive charge and negative charge are moved by the opposite direction towards each other. C. The thickness of the boundary. D. Temperature. Diffusion is driven by the kinetic energy of the particles, and temperature is a measure of that kinetic energy. The warmer a substance is, the more rapidly its particles diffuse. This is why sugar diffuses more quickly through hot tea than through iced tea. E. Membrane permeability. Diffusion through a membrane depends on how permeable it is to the particles. Cells can adjust their permeability to such a substance by adding channel protein to the membrane or taking them away. This means the cell create these channel proteins according to its need for the certain substances. 2. Solvent drag. The second force producing movement of the substances between the body compartment, that's called the solvent drag. When solvent is moving in one direction across the cell membrane, the solvent tends to drag along some molecules of solute. This force is called solvent drag. In most situations in the body, its effects are very small. That means the drug substances with the solvent doesn't have a major effect on the function of the cell. 3. Filtration. Filtration is the process by which a fluid passed through an epithelial layer or other barrier due to a difference in hydrostatic pressure on the two sides. And all of you know that the filtration process, we mentioned to it previously by the renal system. Filtration is affected by some factors. A. The pressure gradient across the membrane. B. The surface area of the membrane. C. The diameter of the membrane pores. D. The size of the filtered molecules. In physiology, the most important case of filtration is seen in the blood capillaries, where blood pressure forces the fluid through gaps in the capillary wall. This is how water, salts, nutrients, and other solutes are transferred from the bloodstream to the tissue fluids, and how the kidneys filter waste from the blood. Capillaries hold back larger particles such as blood cells and proteins. This figure showing the channel proteins, receptor proteins, and recognition proteins that play a major role in transporting the substances from into and out of the cell. 4. Osmosis, osmolarity, and osmotic pressure of the body fluids. Also, all of you know about the osmosis, which is specific for water transport. The osmosis is the flow of water solvent molecules across a semipermeable membrane through channel protein, especially those called aquaporins, into a region in which there is a high concentration of a solute to which the membrane is permeable. Cells can regulate the rate of osmosis by adding aquaporins to the plasma membrane or removing them. Certain cells of the kidney, for example, install or take away aquaporins to regulate the rate of water loss from the body in the urine. The osmolarity is the concentration of osmotically active particles, the particles which attach to water to it, the particles which attach water to it in a solution. The osmotic pressure is an attractive force of the solute particles that drives water molecules to move from solution of low concentration of solutes to solution of high concentration of solutes. That's mean the water movement from the high concentration of water to the low concentration of water. The number of the solute particles determines the magnitude of the osmotic pressure of the solution in which it's dissolved. The term tonicity is used to describe the effective osmotic pressure solution relative to plasma in which the normal body cells can be placed without causing either swelling or shrinking. A solution with effective osmotic pressure as a plasma is said to be isotonic, which corresponds to 0.9% solution of NaCl or a 5% of a glucose solution. Those solutions with greater pressure are hypertonic, in which the normal body cells shrink when they are placed in it. Those solutions with lesser pressure are hypotonic, in which the normal body cells swell when they are placed in it. If water is added to the extracellular fluid by injection into the bloodstream, by injection beneath the skin, or by ingesting water followed by absorption from the gastrointestinal tract into the blood, The water dilutes the extracellular fluids, causing it to become hypotonic with respect to the interstitial and to the intracellular fluids. Osmosis begins immediately at the capillary and the cell membranes, with large amounts of water passing to the interstitial and to the interior of the cells. Within a few minutes, the water becomes distributed almost evenly amongst all the extracellular and intracellular fluid compartments. And all compartments will have the same osmolarities, that's to say, the same tonicity. Excess water intake and dilution of extracellular fluid called overhydration or water intoxication. If water is lost from the body by evaporation from the skin, evaporation from the lungs, or excretion of a very dilute urine as in diabetes insipidus, or loss in feces as in diarrhea, the water will leave the extracellular fluid compartment, causing this compartment to have a hypertonic fluid in respect to the fluids in the intracellular compartments. Osmosis begins immediately at the cell membrane, with water passing to the interstitium and plasma and distributed uniformly between the three compartments so that all the three compartments will have the same osmolarities. The overall effect is called dehydration. This figure illustrated the movement of water by osmosis force from the tube B, which contain high concentration of water, to tube A, which contain low concentration of water. So water moves by osmosis into the more concentrated solution until concentrations are equal. 5. Non-ionic diffusion. Some weak acids and bases are quite soluble in cell membranes in the undissociated form, whereas in the ionic form they are cross-membrane with difficulty. Consequently, if molecules of the undissociated substances diffuse from one side of the membrane to the other and then dissociate. 6. Carrier-mediated transport. It is the transport of substances across the cell membrane mediated by a carrier protein, which is found on the surface of the plasma membrane of each cell. This type of transport is characterized by the following. Number 1, stereospecificity. For example, D-glucose is transported by phosphorylated diffusion, but the L-isomer is not. 2. Saturation. The transport rate increases as the concentration of the solute increases until the carrier are saturated. The transport maximum, or Tm. 3. Competition. Structurally related solutes compete for transport sites on carrier molecules. For example, galactose is a competitive inhibitor of glucose transport in the small intestine. Carrier-mediated transport are of two main types. A. Phosphorylated diffusion, when the carrier-mediated transport occurs according to electrochemical gradient and the energy is not required. Passive. The process is called phosphorylated diffusion. It is also called a uniported transport because the transport occurs in one direction. Example of phosphorylated diffusion is glucose transport in muscle and adipose cells is carrier-mediated according to the electrochemical gradient and is inhibited by sugars such as galactose. Therefore, it is categorized as phosphorylated diffusion. In diabetes mellitus, glucose uptake by the muscles and adipose cells is impaired because the carriers for phosphorylated diffusion of glucose require insulin. Here, this figure showing the transport of glucose by a carrier-mediated protein, which it is opened by the action of insulin. And the characterization of this transport is called a uniport transport because the transportation occurs on one direction. B. Active transport is the second form of the carrier-mediated transport. When the carrier-mediated transport occurs against electrochemical gradient, this is opposite to the fusilated diffusion because fusilated diffusion transport occurs with the chemical gradient, and this active transport occurs against electrochemical gradient. That means the substances transport from the region with low concentration to the region with high concentration. The process requires energy, and it is referred to as active transport. Active transport can be divided into two types. The first type is a primary active transport, in which high-energy phosphate compound ATP provides directly to the energy required for the transport process. Such a transport are Na potassium ATPase pump, hydrogen ATPase pump, proton pump, and calcium ATPase pump. This figure showing the active transport for the two substances, potassium and sodium. They transported at the opposite direction or against the chemical gradient from region with low concentration to the region of high concentration by co-transporter protein at the opposite direction. So it's called antiport transport because the two molecules are transported into the opposite direction. Secondary active transport, in which the transport of one substance, for example sodium, according to electrochemical gradients, provides the energy to transport another substance against its electrochemical gradient. The metabolic energy is not provided directly but indirectly from the sodium gradient, which is maintained across the cell membranes by sodium-potassium ATPase pump. Thus, inhibition of sodium-potassium ATPase pump will decrease the transport of sodium out of cell, decrease the transmembrane sodium gradient, and eventually inhibit secondary active transport. Here I have mentioned these types of transport previously, so all of you knows these types of transport. The direction of the carrier-mediated transport is a uniport transport. If one transported substance is moving in one direction and without an associated transport of another substance, as in the fusilated diffusion of glucose, this is called that uniport transport. B. Co-transport or symport, if two or more of the transported substances are moving in the same direction, as in sodium co-transport of glucose and amino acids, sodium potassium 2-chloride co-transport that occurs in the renal system. This is called as co-transport or symport. C. Counter-transport or antiport, if two or more of the transported substances are moving in the opposite direction, as it occurs in sodium-calcium counter-transport and sodium-hydrogen counter-transport. Here this figure showing the forms of the direction of a transporter protein, which it is symport and antiport. In symport, the two molecules carried or transported by the same transporter protein on the same direction. In antiport transport, the two substances are carried in the opposite direction. Dear pupils, At this slide, I'd like to finish my lecture and complete the other slides on the next lecture if God will, with my best regards.